Now I am going to do a role play of an enzyme, restrictions enzyme.
My name is restriction enzyme. I am produced chiefly by certain bacteria, and I have the property of cleaving DNA molecules that are near a specific sequence of bases.
Now I will say about my introductions. I am a restrictions enzyme. I am a protein isolated from a bacteria that cleaves DNA sequences at sequence specific sites and producing DNA fragments with a known sequence at each end. My use is critical to certain laboratory methods including recombinant DNA technology and the genetic engineering. I am incredibly cool and there are at least 3000 of us. Each of us cuts a specific DNA sequence and doesn't discriminate as to where the DNA comes from, either from bacteria or it came from fungi or mouse or human being. We'll cut it in a snip in a small pieces in all DNA. Now I will say about my history, how I was discovered and how I was characterized. I was first discovered and characterized in late 1960s and early 1970s by molecular biologists Werner Arver, Hamilton O. Smith and Daniel Nathans. My ability as the enzymes to cut DNA at a precise location enabled researchers to isolate gene containing fragments and recombine them with other molecule of DNA, that is to clone genes. My name as a restriction enzymes are derived from the genus, species and strain designation of the bacteria that produce them. For example, the enzyme EcoR1 is produced by the Escherichia coli strain RY13. Here E is giving the genus name, CO is giving second two letter middle name is giving the species name, and RY is giving the strain in which it is discovered. Like now I will say about my classification. So we means restriction enzymes are commonly classified into 5 types. On the basis of what? On the basis of in different structures, or where we cut the DNA substrate at the recognition sites, or if the recognition and cleave sites are separate from one another. And types are type 1 enzyme, type 2 enzyme, type 3 enzyme, type 4 enzyme and type 5 enzymes. Type 1 enzymes mean their EC number is EC 3.1.2.1.3, and we will discuss EC number in upcoming will in later 2 minutes later or 1 minute later. And type 1 enzymes cleaves at the sites remote from recognition sites, require both ATP and S-adenyl adenosyl L-methionate to functions. Multifunctional protein with both restriction, digestion and methylase activities. Type 2 enzymes cleave within or at short specific distance and distance from recognition sites, must require magnesium single functions enzyme independent of the methylase. Type 4 enzymes target modified DNA, that is methylated hydroxy, methylated glucosyl hydroxy methylate DNA. While cutting will modify the DNA by adding the methyl group by hydroxyl groups, so to reduce their further reactions. Type 4 enzyme utilize guide RNAs, means it's one type of the RNA, that is guide RNA, that is utilized by the type 4 enzyme. Broadly we are 2 types, that is according to the nucleases activities, restriction endonuclease and restriction exonuclease. Endonuclease means will cut the DNA at in between in middle of the strand. Exonuclease means will cut the DNA at the end. While discussing about my history, I explained the term that is EcoR1. That is now we will see in detail about the EcoR1, what is name, why it came, and it is come under the my nomenclature. So first, since my discovery in 1970s, many of us restriction enzyme have been identified. For example, more than 3500 different type 2 restriction enzyme have been characterized. Each of us is named after the bacterium from which it was isolated. Means if it is isolated from the Escherichia coli, then my name will be given as a EcoR1. If it is from the any bacteria, from that bacteria genus name and the species name, my name is identified using a naming system based on the bacterial genus, species and strain. First word will be the genus, second two middle two word will be the species, and the last two will be the strain, and 1 2 3 will be the in which order it is discovered. So example is given of E. coli. E means strategy, that is genus. CO means coli, specific species. R means RY13, that is a strain of the E. coli, in which strain of the E. coli it is discovered, and 1 it is a order of identification in the bacterium. While discussing the types of classification, my classifications, I said about the enzyme commission nomenclature, that is some term of type 1 came EC 3.1.21.3. now we will discuss about how it is came. EC means enzyme commissions, which gave the actual nomenclature of the enzyme. So EC refers to the enzyme combination. 3 refers to the classes. We know that there are the 6 classes of the enzyme, that is oxidoreductase, transferase, hydrolyses, ligases, like that there is 6 classes. In this order, 3 is the subclass name, that is a hydrolyses, and 1 is the subclass, the class that comes under the hydrolyses, that is the 1 means acting on the ester bond, that is and 2 1 is a subclasses. 2 1 refers to indo-deoxyribonucleases producing 5-phosphomonoester, and last word 3 is the type, means in which order it is falls. So it means that this enzyme type 1 act on the ester bond according to the subclass, and it comes under the class of the hydrolyses. In introduction part, I said about myself that I will act on the recognition sites at a particular sites. According to that only I am divided into either endonucleus or exonucleus. Now we will discuss about the in which recognition sites I will react. So we means restriction enzyme cut DNA bonds between 3' OH of one nucleotide and 5' phosphate of the next one at the specific restriction site. That site is a palindromic sequence, means from both sides it will come same. Adding methyl group to certain bases at the recognition sites on the bacterial DNA block the restriction enzymes to bind and protect the bacterial DNA from being cut by themselves. Each of us restrictions, that is restriction enzyme, recognize a short specific sequence of nucleotide bases. These regions are called as recognition sequence or recognition sites and are randomly distributed throughout the DNA. Different bacterial species make restriction enzyme that recognize different nucleotide sequences. A straight cut of us generates a blunt ends where both strand terminate in base pair. Blunt end are also called non-cohesive ends since there is a no unpaired DNA strand fleeting at the end of DNA. We in upcoming session we will discuss about what is the blunt end, how it is cut, in mechanism we will discuss everything. Now only I said about that I will cut at the specific sequences, that is the palindromic sequences. Now here we search in gene, there is a sites where there will be the specific sequences that will end and start with the if we will opposite this means same word will come, that is like a madam, madam. If we start from end or end, or we will start from starting point or the ending point, it is come the same word. Like that in whole gene sequences it will identify the specific palindromic sequences and cut at that specific sequences. Here T T A G C A C on 5' to 3' sequences it's identified, and in the and it complementary sequence strand, that is 3' to 5', there also there is a C palindromic sequence of T T A G C A C C A C G A T T is there. So whenever palindromic sequences came, it identified their respective palindromic sequence and cut the DNA at the respective site. Now I will explain about my mechanism of action, means how I will act and what will be the significance of my mechanism. Here is the example of two gene. one is original DNA molecules and other is a foreign DNA molecules. And one restriction enzyme is there, because in both gene there is a same sequence, same palindromic sequences. So only one type of me got these DNA sequences at a specific site, that is a GA of the both. Between GA nucleotides bond will be broken and two fragment of the DNA molecules will forms, and that is in second steps. After that, with the help of DNA ligase, both the fragments is stick together, while after the breaking the bond between the GA, two fragments are the forms, and both the fragments have the sticky end, that is a sticky end, and that sticky end will join together by the DNA ligase, and it will form the other recombinant DNA molecule. Now let's see how inhibitor EcoR1 methylase inhibit my activity. In first case, there is no any masking in the DNA sequences. Restriction, I will go and I will cleave the DNA sequences at a specific palindromic sequence recognition sites. So in second case, there is in first step EcoR1 methylase is coming and it is masking my binding site. So I will not cut the DNA sequences at the palindromic sequences. Thus the inhibitor is affecting my DNA, my activity. Here I will say how I will cleave the bond between two nucleotide. Both two nucleotide is joined together by the phosphodiester bond. I will go and bind with the phosphate group of the phosphorus of the phosphate group, and it leads to the release of the H plus ion from the molecule, and one of the nucleotide make the linkage with the magnesium ion, and one with the nucleotide takes the phosphate group. Thus it leads to conclusion that one of the nucleotide will make the base pairing with the other fragments of the DNA. Thus it is making one end as a blunt end and other as a sticky. Now I said how I will cleave the DNA of the one types, means in one straight DNA. But now I will say about the how I will cut in the bacterial DNA, that is a plasmid. Both foreign DNA, that is from the eukaryotic, and another is plasmid from the prokaryotic. So restriction enzymes will cut both the DNA, foreign DNA and the plasmid DNA, one from the eukaryotic and another from the in the same base, both plasmid and the eukaryotic DNA, I will recognize the specific recognition sites, and I will cut at both the sites. In recombination DNA technology, foreign DNA is joined to the plasmid DNA and it will form the recombinant plasmid DNA, and it leads to the production of the many antibiotics and many valuable products. During nomenclature, I said that according to from which bacteria I was isolated, according to that my name was given. If I am isolated from the Escherichia coli, then my name is given E. coli. If I am isolated from Thermus aquaticus, then my name is given as a TAC. If I am isolated from Haemophilus influenza, my name is given HIN. Like this basis only. There are the many examples of the restriction in my examples. Now I will give my examples and from where I am, from which organisms I am isolated, and what is my recognition sequences. So first of my example is EcoR1, that is Escherichia coli, and strain is R, and order of identification is I, and I will cut the DNA sequences between the GA and GA should be present in the DNA sequence of the GAATTC, and my next example is a TAC1, Thermus aquaticus. T is the genus name and A is a species name. I is a order of identification. I will identify TC sequence and I will cut between the TC nucleotides. Third example is a HIND3. As for in Haemophilus, genus name IND for the influenza, that is the species name, and 3 is the order of identification. And hint 3 will identify the nucleotide sequences, that is AA, and cut between these two nucleotide sequences. So fourth example is BAM-H1. B refers to the bacillus, AM refers to the amyloid coefficients, and H is strain name, and I refers to the order of identification. And BAM-H1 identifies sequences GC and cut between these two nucleotides. Fifth example is ALU1. A refers to the Athrobacter, and LU refers to the luteus, and I refers to the order of identification. And ALU1 identify the GC. ECO R1 GA and ALU1 GC, in between AGCT palindromic sequences. After discussing about my introduction, my history, my nomenclature, and my mechanism of action, how I am acting, how I am cutting DNA strand by recognizing a specific palindromic sequences, and my examples according to how I am my name is given according to the bacteria from where it is isolated. Now I will say my uses, how I am useful in various sector. Nowadays in every field of the biotechnology, restrictions enzyme, that is I am useful. Without me, no any genetic engineering work can be done. If anyone have to do genetic modification of the plant or animal, then there should be the my uses. If anyone want to do the gene therapy, then they have to use me. Without me, they can't do gene therapy. If there is isolation, if there is a gene manipulation to obtain the various different product, then there should be uses of me. Without me, they can't cut the DNA and they can't use it for further use. In southern blot technology, to digest the genomic DNA for gene analysis, I am useful. I am also be used to distinguish gene allele by specifically recognizing single base changes in DNA known as a single nucleotide polymorphisms, SNPs. Not only this application in plant biotechnology, in enzymology, in immunology, I am very useful. In diagnosis field, in PCR technology, in ELISA technology,